Welcome and thank you for joining us today for webinar Accelerating Lead Identification through Integrated Antibody Development and Characterization Workflows. My name is Jean-François Belec. I'm a Market Development Manager at Bio-Techne, and I'm very excited to welcome you to today's event. So I want to just start by a big thank you to you, audience, for joining us today. We will start with a short introduction, some housekeeping items that I'm sure you're very familiar with if you listen to other webinars. And then we have three presentations from respectively Mike Heitman, Jessica Fij, and Srinivasa Rao. And it will be followed by a live Q&A to answer any questions that you may have. So that's me to encourage you to check out your resources section as well. Where you can find a lot of content that is related to today's presentations, including the Maurice product brochure and two application notes, which are demonstrating the workflows that we will be talking about today. On this slide, you have a quick overview of the topics that we will cover, and that will be covered by Matt, Jessica, and Srini. And also, you may have noticed recently that we've changed things a little bit at Bio-Techne. R&D Systems, which is a brand that is very well known in the life science industry, is now a portfolio brand. And what does that mean? It means that we're bringing together the tools that researchers have long relied on within the same brand. So, of course, it includes all the R&D systems, reagents, and kits that many of you are familiar with. But also now the analytical instruments that used to be protein simple, and as well as top small molecules, novice antibodies, and many more, all under one name, one family, R&D Systems by Bio-Techne. What does that mean practically for you? That means that protein simple, if you're a user of one of our analytical instruments, is now R&D Systems by Bio-Techne. And the brand name may have changed, but you'll still get the same teams, the same support, and the same drive to make innovative products. And now I'd like to introduce our first speaker, Dr. Matt Heitman. Matt is a manager in the Scale and Purification Group within the global R&D team at Bio-Techne, and he's focusing on antibody discovery efforts. He leads a specialized team focusing on cell culture, purification, stable cell line development, and the biophysical characterization of high-quality antibodies. These reagents serve as the backbone for clinical advancements in diagnostics, in cell and gene therapy, and in spatial biology. Prior to joining Bio-Techne, Matt was a senior scientist at Glycogen, where he specialized in microbial engineering and the production of bioactive oligosaccharides. He holds a PhD from Dartmouth College, and he completed his postdoctoral fellowship at Tufts University. I think I've said enough. So Matt, take it away.
Great, thanks for that introduction. So the title of my talk today is Recombinant Antibody Production and Analysis. So before I get into the data portion of my talk, I'd like to talk a little bit about Bio-Techne and who we are as a company. So Bio-Techne is a Minneapolis-based global life sciences company that develops and manufactures high-quality reagents, instruments, and diagnostic tools. And these reagents are really designed to support researchers in academia, biopharma, as well as in the clinical diagnostic markets. And our mission is to improve the quality of life by catalyzing advances in life science and medicine. And this slide really highlights or summarizes the power of Bio-Techne in that we have over 750,000 quality products. We are 3,000 committed employees worldwide, and we have 45 years of innovation experience. And our products have been attributed by over 500,000 user citations and 7 innovative instrumentation platforms. Furthermore, we are a GMP certified company, so you can expect consistent high quality from us every time you engage with us. And so some examples of our products are shown on this slide. For example, in the cell culture, media, recombinant cytokines, as well as bioreactors to support immune cell therapy. And so Bio-Techne combines the quality and legacy of three high-quality companies, R&D Systems, Novus Biologicals, and Tokris Biosciences, into one innovative reagent solutions provider. As I mentioned before, our mission is really to provide researchers worldwide with a comprehensive range of proteins, antibodies, small molecules, and immunoassays, which have been developed over decades by our skilled scientists. And so whether you're an academic researcher working to unravel the mysteries of biology, or you're developing a therapeutic, scientists can rely on our products and confidently push the boundaries of their experiments to achieve their next breakthrough. On this next slide, I'd like to briefly spend a little more time highlighting the history of Bio-Techne. Specifically, we were founded as R&D Systems, an established leader for RUO recombinant proteins and ELISAs, in Minneapolis in 1976, and the company was renamed as Bio-Techne in 2014. We've shown continuous growth for over 45 years, and along the way, we've acquired several reputable companies, allowing us to broaden our portfolio and the extent by which we can support scientists in their goals. We've had several acquisitions, with a company called Lunaphore being the latest. And this company provides a cutting-edge instrument for spatial biology applications, where researchers can interrogate a tissue section with multiplex panels of antibodies to examine biomarkers simultaneously in the same tissue sample. So now I'd like to dive into the more data-oriented portion of my talk, which will focus on recombinant antibodies and how the Maurice instrument is a valuable tool for their characterization early in the development workflow. And on this slide, I'd like to provide an introduction to recombinant antibodies, which are antibodies that are produced through the use of recombinant DNA and in vitro expression systems, rather than through traditional animal immunization. So that's highlighted down here on this graphic, where basically the sequence of your antibody is known in the form of recombinant DNA, and that's portable and can be used in any expression system that you desire, whether it be CHO, HEC, transient, or stable. And so the value and key uses of recombinant antibodies are many. They have high value as research tools, for example, as flow reagents or for Western blotting. They can also be highly valuable in the cell therapy space as selection or activation reagents. They have high value as high affinity binders in diagnostics and immunoassays. And furthermore, most antibody therapies are in fact recombinant antibodies. And so recombinant antibodies offer many advantages over traditional animal-based immunization that I've highlighted up here in this box on this slide. Particularly in manufacturing, recombinant antibodies offer more consistency in your manufacturing process, in that they're not subject to genetic drift that can be an issue with hybridoma-based expression systems. Furthermore, recombinant antibodies are scalable in that you can make many of them in high throughput at small scale, or if you need large quantities, you can generate a stable cell line, which can be scaled to many thousands of liters to generate grand quantities of an antibody that is of interest. And finally, since the sequence of your antibody is known and there is higher degree of consistency in the manufacturing process, this provides more solid long-term supply for an antibody or set of antibodies of interest. And finally, recombinant antibodies are highly amenable to high throughput production platforms, particularly in a transient system that I'll highlight on the next few slides. In addition, recombinant antibodies offer a lot of opportunities for engineering to modify an antibody to fit a particular need, again, which is not really possible when you're talking about a hybridoma-based expression system. So since the sequence is known, changes of interest can readily be imparted, such as isotype or species swaps. An antibody of interest can be reformatted if that works better in a particular assay of interest. For example, an antibody can be converted to a Fab or a single chain or bispecific format. And finally, in the age of AI and powerful engineering tools, antibodies can be subject to affinity improvements as well as stability improvements, and that really relies on knowing the starting sequence. However, with recombinant antibodies, there are, of course, challenges. So in an era where you can make many variants of a particular antibody, how do you establish or generate a robust pipeline that enables high throughput production? And in addition, what tools are needed for scalability to generate stable cell lines to produce high mass of a particular antibody? And finally, as we're learning more about antibody structure and sequence and developability concerns, what are some robust analytical tools that can be brought to bear early in the process? And so to address challenges with high throughput production of antibodies, we've developed a high throughput antibody expression and purification platform that enables generation of 100 to 200 purified antibody preps over a 10-day time period. And so specifically, proprietary expression plasmids harboring the antibody sequence of interest are transiently transfected into suspension-grown HEC293 cells, as shown here in this graphic. After 7 days, culture media containing the secreted antibodies of interest are subjected to purification using semi-automated purification instruments. And then purified antibodies are exchanged into a standard buffer and are ready for testing. So as I mentioned before, at Bio-Techne, we use this workflow to produce antibodies for screening in a variety of applications, including immunohistochemistry, multiplex immunofluorescence, ELISA, as well as simple Western. And so typically to assess the quality of the antibodies that we produce in our development workflows, we run SDS-Page to assess purity, as well as leverage techniques such as SEC-MALS to determine antibody aggregation and purity. However, as I want to highlight in this text or in this talk, antibodies that we produce in this workflow in large scale and high throughput can also be analyzed by the Maurice instrument, which provides information on size, purity, and charge heterogeneity, as I've highlighted up here on this workflow. And so these are really important physical properties that are worth measuring early in the candidate selection process, especially when screening therapeutic candidates where developability is critical. And so later in the talk, my colleague Srini will show how recombinant antibodies produced in this workflow were also analyzed on the Maurice and how this analysis identified some features that could be important for developability going forward. And in addition, as I mentioned before, recombinant antibodies can also be used to generate stable cell lines if you need to grow up large quantities of cells to make high mass quantities of an antibody of interest. And that workflow is shown here, basically where the antibody sequence is incorporated into the genome of a host cell of interest. Single cells, candidates are grown out of that stable pool, and then those can be screened and scaled. And early in that clone selection process, the Maurice can also be used to identify candidates that may have issues or may have properties that are desirable going forward. So as I mentioned on the prior slide, another key advantage of recombinant antibodies, in addition to being amenable to high throughput production, is their scalability if you need large quantities of an antibody of interest. And so once the sequence of your antibody is known, that's very portable, and a stable cell line can be generated for large-scale production of your antibody of interest. And so stable cell lines can be grown in culture volumes from tens to hundreds to thousands of liters, and theoretically enabling gram-scale production of your antibody of interest. And this is really key for initiatives where high mass amounts of a desired antibody is needed, as would be the case for possibly developing a therapeutic antibody. And so that general workflow is shown here, where a stable cell line can be grown in various cell culture formats. The antibody is purified from the conditioned media, and finally testing in QC occurs. And so a key part of that, of course, is having a robust, high-performing stable cell line where the antibody DNA sequence has been chromosomally integrated and stably produced. And so Bio-Techne has a key technology called TC Buster that allows for generation of highly productive stable cell lines. And TC Buster is a transposon system. This enables multi-copy genomic integration of your antibody sequence of interest into a host cell, such as CHO or HEC, via a cut and paste mechanism, whereby the coding sequence for your antibody of interest is cut out of the expression vector by the TC Buster transposase and then pasted into the host cell chromosome. And so later in the talk, you'll hear from my colleague Jess Fige as to how TC Buster can be used to generate highly productive CHO stable cell lines, and furthermore, how the Maurice instrument was a powerful analytical tool that enabled good selection of clones early in that development process. And so in my last slide, I'd like to highlight that in the development of recombinant antibodies, biophysical characterization of candidates is critical early in the screening process. And this is particularly true in the therapeutic antibody space, where molecules have a high bar, not only for their efficacy, but also in manufacturability and stability. And so some of those parameters are shown here. So for example, for any therapeutic antibody, you'd want to know that it binds your target, of course, but you also want to know that there's minimal off-target activity. You want to have information regarding structural stability and solubility, as this connects to properties such as thermostability and propensity for aggregation. And you also want to have information regarding charge heterogeneity of your molecule, as this connects to concerns or decisions relating PI and formulation, stability and solubility of your molecule, as well as pharmacokinetic concerns. And so Bio-Techne offers a powerful instrument called the Maurice, shown here, that provides some information regarding some of these biophysical considerations. And so the Maurice can be run in two modes. First, it can be run in capillary electrophoresis SDS mode, and this provides information regarding size and purity of your antibody molecule. So antibodies can be separated by size, and those different size fragments or species can be quantitated. And in this mode, the Maurice can be run using non-reducing and reducing conditions. And secondly, the Maurice can be run in IC-IEF mode, which is imaged capillary isoelectric focusing. And in this mode, the instrument provides information regarding charge heterogeneity and PI of your molecule, which are very important to know early on in the screening process. And so again, as shown in this graphic, charge variance relating to a particular antibody can be separated, and those peaks can be quantitated. And so in the next portion of the talk, you're going to hear about exciting work where the Maurice provided valuable upstream information for antibodies that are produced in the high throughput transient platform I described, as well as from stable cell lines generated using TC Buster technology. So with that, I'll end my portion of the talk and hand it back over to our moderator. Thanks.
Thank you so much, Matt, for this great presentation. And now it's my time to introduce our next speaker, Dr. Jessica Fige. Jessica is a scientist specializing in cell and gene therapy with deep expertise in immune cell engineering and therapeutic development. She earned a PhD in immunology from the University of Minnesota, where she applied cell editing technologies to investigate T-cell activation and differentiation, followed by a postdoctoral research focused on innate and adaptive immune responses to viral infection. Jessica is currently the supervisor of the immune cell therapy team at Bio-Techne, where she leads efforts to advance non-viral gene editing for immunotherapy and bioprocessing applications. And in her role, she oversees projects aimed at improving immunotherapy manufacturing with a particular focus on developing high quality and cost-effective treatments. Jessica, the floor is yours.
Thank you for that introduction. I'm really excited to talk to you today about how we use the TC Buster transposon system to edit CHOK1 cells to make better antibody-producing clones with higher titers and higher copy numbers. So for those of you who haven't heard about TC Buster, it is a transposon system that can non-virally insert cargo into your cells of interest. So the TC Buster N is a hyperactive transposase that works great in mammalian cells. So to use TC Buster, you need three things. The first of which is an electroporator. So that's going to deliver the TC Buster components inside your cell. You need the mRNA that encodes the TC Buster enzyme, and you need a plasmid transposon that encodes your gene of interest. That transposon needs to be compatible with TC Buster, meaning that it has the TC Buster specific ITRs flanking your gene of interest. So those materials are delivered by electroporation. Once inside the cell, the mRNA is translated into the transposase enzyme that binds to the ITRs flanking your gene of interest. TC Buster cuts this cassette out of the plasmid and inserts that randomly into the DNA, where you get stable long-term expression of your gene of interest. So we've used TC Buster in a variety of cell types with a variety of cargo sizes. So if you are interested in learning more, we have tons to share, but we've used TC Buster in T cells, NK cells, iPSCs, CHO K1 cells, which I'll show today, as well as HEX cells. If you are interested in knocking out a gene at the same time, you could use TC Buster in conjunction with CRISPR-Cas9 to knock out that gene of interest in the same electroporation step. To dive into what we did today, we first took our CHO K1 cells and edited them with or without TC Buster and a transposon encoding CD28 antibody. We then selected them to create stable pools, analyzed those stable pools, and then moved on to single cell cloning, analysis of those clones, and then performed a quick screen at a more manufacturing scalable level. So we're going to focus the first part on how we made the cells and just analysis of those stable pools. So to do this, as I just mentioned, we had CD28 antibody in a transposon. So that cassette flanked by the ITRs has a promoter, the CD28 antibody, and a puromycin resistance gene. We use that to aid us in selection after editing. We had two conditions that we were testing with these CHO K1 cells. We either had random integration alone, where we only delivered the transposon via electroporation, so similar to how historically cells have been made to be antibody producers, or we added the TC Buster mRNA to the electroporation reaction to aid in that genome insertion. So once we had performed the electroporation reaction, we then allowed the cells to recover for 2 days before putting them on puromycin for selection. So what you're looking at here is about a month of growth of the CHO K1 cells after editing. So you can see in the random integration, the dark blue, as you're going along over the weeks, you're just starting to see those cells start to recover at around 22, 23 days. So they would probably need another week or so before you would have a stable pool population large enough to be able to assess them. Whereas when you look at the TC Buster integration, you can see those cells start to recover around 10 days. And then just before 20 days, they really start to take off. So those pools respond quicker, and you can generate those stable pools 1 week sooner with TC Buster immediate generation, as opposed to just random integration. So now that we had our stable pools, we wanted to assess them for the ability to secrete the CD28 antibody, as well as understand how many copies per cell were present in these pools. So we took these cells and put them in a static 6-well plate for 7 days. At the end of 7 days, we harvested the supernatants to look at the presence of the antibody, and we harvested the cell pellets to look at the copies per cell. So starting over here on the left, looking at the antibody titer. So we did a IgG ELISA, looking for the presence of the CD28 antibody in the supernatant. We set the random integration at 1, and we were able to see a 5-fold increase in the amount of antibodies produced in the TC Buster pool compared to the random pool. Then looking at the copy number, we ran digital PCR, looking at the inserts in the genome of these pools. So the random integration pool had 1 to 2 copies in the pool, whereas in the TC Buster pool, we were seeing 6 to 7 copies. So even at this early stage, just looking at the stable pools, we're seeing 5 times more antibody and 4 times more copies per cell. So now that we have strong stable pools, we wanted to proceed to single cell cloning and then analyzing those cells once again for secretion of that antibody and copy number. For single cell cloning, we took each of our pools, the random integration and the TcBuster integrated, and we seeded them a single cell per well in two 96-well plates. We let them to recover for a couple weeks, monitoring them, and we were able to pick 58 clones from the random integration pool. And TcBuster pool, we had 72 clones. So we were able to pick more clones after TcBuster editing compared to random integration. So we screened a number of those clones and then proceeded to take the top 3 highest producing clones and move them to 125 ml shaker flasks. In the shaker flasks, we allowed them to grow for 10 days and then harvested those supernatants to look at ELISA again for the presence of the CD28 antibody and repeated the copy number. So for each of these graphs on the far left, we're comparing back to the pool and then each of the clones. So you can see here on the far left, looking at the antibody titer, the pool for the random integration is producing less than 100 mg per liter of the CD28 antibody, and the 3 clones are similar. Once looking at the TcBuster integrated pool, you can see the pool as a whole on this 125 ml shaker flask production, we're producing around 500 mg per liter of the CD28 antibody. One of the clones was a little bit lower, around 400 mg per liter, but the other 2 clones were at 800 and almost 1 gram per liter. So these clones are producing significantly more antibodies than the random integration clones. We also looked at the copy number. You can see the pool for the random integration as well as the 3 random integration clones have 2 or less copies, whereas the pool and the 3 clones for TcBuster had 4 or more copies, with clone 3 having 8 copies. So we're having a higher copy number and higher clones coming out of, higher producing antibody clones coming out of the TcBuster editing. The last thing we wanted to do was to screen one of the top producing clones from each group and see if they had manufacturing potential. To do this, we used the Amber 250 bioreactor system. So in the bioreactor system, we had 4 culture conditions for the TcBuster and the random integration clone. These were each run in duplicate. So the 4 conditions were 2 different media types and 2 different temperatures. We're a few days into the protocol, we dropped the temperature to 33 or left it the same as the initial temperature. So at the end of 14 days, we once again ran an IgG ELISA looking for the concentration of the CD28 antibody. So you can see across the 4 culture conditions for the random integration clone that we were producing less than 1 gram per liter. The first 3 culture conditions for the TcBuster clone, we were around 4 to 6 grams per liter. With the last condition, we were over the average of the 2 bioreactors at 12 grams per liter. So in all the conditions that we tested for the TcBuster clone, we were at or above industry standards, with one of them at 12 grams per liter. So to summarize what I've shown you here today, using TcBuster, you're able to get to your stable cell pools faster. Those pools are going to be indicative of the clones that you generate. The clones with TcBuster had higher titer clones with more copies per cell, decreasing the number of clones that you will likely need to screen to get to your final product. We ran a quick screen on the Amber 250 and was able to quickly identify a cell culture condition with 12 grams per liter. So this decreases the amount of screening time you need to be able to achieve those industry standard yields. If you are interested in learning more about TcBuster or any of the data that we share here today, please use one of the QR codes here to link you to the TcBuster webpage or to the app note. With that, I'm happy to hand it back to our host. Thank you.
Thank you so much, Jessica, for introducing TcBuster to our audience. Non-viral alternative are very, very much in demand. So it's great to see this innovation from Bio-Techne. And it's my pleasure to introduce our final speaker, Dr. Srinivasa Rao, who joined Bio-Techne in July 2022 as a staff scientist. Prior to that, he gained extensive experience at other biotech companies, including Thermo Fisher Scientific, where he helped to develop industry-leading HPLC columns that separate biomolecules by size and charge, and at PhenomenX, which is now a Danaher company, where he contributed significantly to separations and new column development and their applications. He has a PhD in biochemistry from Osmania University in India and he followed this by his postdoctoral studies at NYU and Albert Einstein College of Medicine. I think I've said enough, so it's time to hand it over to Srini.
Good morning, everyone. This presentation focuses on high-throughput antibody analysis using Morris platforms. We highlight how integrating analytics with antibody production accelerates lead selection. The objective is to demonstrate faster data-driven decision-making. Bio-Techne's mission is shown here. We improve the quality of life by catalyzing advances in science and medicine. Our platforms enable generation of high-quality, reproducible data. These capabilities support both discovery and development workflows. Morris and Morris Flex cartridges are shown here. These provide rapid CE-based analysis for protein characterization. Pre-assembled cartridges enable reproducibility and ease of use. Methods can be seamlessly transferred across development stages. Morris integrates charge and size analysis on a single platform. This reduces capital investment while expanding analytical capability. Automated workflows ensure consistent and compliant results. ICIEF enables high-resolution charge heterogeneity analysis. Real-time focusing allows rapid method optimization and troubleshooting. Quantitative data can be generated within few minutes. You can easily develop methods in a day. The ICIEF technology ensures consistent results for charge variant separation with pre-assembled cartridges and automated sample injections. We offer multiple platforms to meet specific needs. Whole column imaging enables high resolution, high quality data within 10 to 15 minutes. The ease of use and high reproducibility make it a great tool for use in QC. When compared to traditional ion exchange chromatography methods, the Morris and Morris Flex instruments provide better speed and resolution for routine GMP testing with up to 60% in time savings and a 97 to 98% reduction in hazardous waste generated. It's a flexible platform that allows for deeper characterization without additional method development needs. CESDS provides accurate protein size and purity assessment. Automation improves reproducibility compared to gel-based methods. The workflow is fast and eliminates hazardous reagents handling. The CESDS technology provides improved reproducibility with simplified sample prep and automated sample injections. CESDS analyzes samples without any acrylamide and generate considerably less waste than traditional SDS page, providing a greener alternative. It is also faster than SDS page, and you can get high quality quantitative data in as little as 5.5 minutes for each sample. CESDS significantly improves efficiency compared to SDS page. Analysis time and waste are substantially reduced. This makes it ideal for high throughput environments. This visual comparison of the workflows of SDS page and CESDS highlight the significant time savings one gets, along with a massive reduction in waste generation. On an average, gels could take anywhere between 3 to 7 hours, while you can get results through CESDS in as few as 2 hours, depending on the number of samples you analyze. Let's look at HTP mAb analysis workflow. This workflow integrates antibody production with downstream analytics. High-throughput processes enable rapid generation and testing of candidates. This ensures timely data for decision-making. We integrated a high-throughput recombinant antibody platform with capillary electrophoresis on the Morris system. We produced and purified over 100 recombinant antibodies for this analysis. We evaluated charge heterogeneity using supersonic ICIEF method with onboard mixing feature. We performed rapid CESDS analysis using turbo CESDS cartridges and with stacking method. Grouped samples into 3 sets to streamline data interpretation. Set 1 CESDS results show predominantly intact antibody in non-reduced conditions, and this reflects purity of the antibody. Reduced analysis confirms expected heavy and light chains. Variability across samples is minimal. But these are different samples and small changes may be significant. CSDS analysis set 2. The results show extensive fragmentation indicating reduced stability. In a standard CSDS run, sample is incubated at 70 degrees centigrade for 10 minutes. Antibodies that are not stable to this treatment show high percentage of fragments with a low percentage of intact peak. CSDS clearly distinguishes these unstable candidates. This enables rapid identification of liabilities. CSDS analysis of set 3 is shown here, and this set 3 displays mostly intact antibody under non-reducing conditions with low fragmentation. Reduced data confirms structural components, both HC and LC components. This set represents relatively stable candidates. With Morris platform, using lane view feature, one can easily compile all the CSDS runs. Comparison across all sets highlights clear stability differences. Non-reduced panel is on the top and reduced panel is shown in the bottom. Heat labile and heat stable antibodies are easily differentiated. Now let's look at the ICIF profiles of all the 3 sets that we just discussed. For ICIF analysis of these antibodies, we used onboard mixing features, OBM. Now, OBM workflow simplifies ICIF operation by minimizing manual intervention that reduces variability. The process is optimized for high throughput and reproducibility. This operational workflow is shown here. Make a 1.25x ampholyte mix, prepare cartridge and reagent, OBM vials, transfer 25 microliters of 5x protein sample into a vial or a plate, load cartridge reagent samples into Morris, enable OBM, and build batch. Hit start. This electropherogram shows a typical separation and labeling of charge variants. Acidic main and basic peaks are clearly resolved. Acidic peaks are left to the main peak, and basic peaks are to the right of the main peak. Set 1 ICIF results show different charge profiles that are based on their properties. Variants are well separated across samples, showing different acidic and basic charge variants. Knowing percent of acidic variants as well as basic variants play an important role in further development of antibodies. Set 2 data reveals shift in charge distribution. Increased variability correlates with instability, and increased percentage of basic variants are observed. Set 3 shows distinct charge behavior with higher basic variants. These differences are important for developability. ICIF enables rapid differentiation between these samples and shows a path for monoclonal antibody development. Summary so far: high throughput production enabled rapid generation and purification of over 100 recombinant monoclonal antibodies. Turbo CSDS and ICIF on the Morris system provided fast, reliable profiling of purity, fragmentation, and charge variants. Integrated workflow combined HTP antibody production with capillary electrophoresis analytics for streamlined candidate evaluation. ICIF and CSDS resolved distinct peaks across all antibodies, reflecting key structural and quality attributes. Set-wise analytical trends supported rapid, informed candidate triage and early selection decisions. We now evaluate reactor supernatant directly on Morris. Morris enables rapid in-process assessment. This accelerates upstream optimization. Jessica already explained the workflow earlier. The pipeline integrates cell engineering and characterization. Each step is designed for scalability and throughput. Analytics guide decision making at each stage. Amber reactor samples and cell culture conditions. CHO clones expressing CD28 mAb. Different media, Dynamis or ActiPro, and different temperatures, 36.5 or 33 degrees centigrade, were screened systematically. This enables efficient exploration of process parameters. Minimal material is required. We compared CSDS profiles as well as ICIF profiles under these varying conditions. Turbo CESDS analysis of reactor supernatant is shown in this slide. Percent peak areas are shown. Top panel shows the non-reduced analysis and the bottom shows the reduced analysis. A representative electropherogram is shown on the left and the data from various reactors are shown. Turbo CESDS enables rapid analysis of supernatants. Both reduced and non-reduced conditions are assessed. This provides quick insights into product quality, including purity and fragmentation. This slide shows CESDS analysis under non-reduced conditions. Peak areas are shown. Reactor 10, which has dynamics media, is compared with reactor 13 active pro media. Both are at 36.5 degrees centigrade. Media comparison shows increasing monomer levels over time for both reactors. CESDS captures these trends quantitatively. This supports media selection. This slide shows non-reduced CESDS analysis, where we did the comparison of reactor 10, which uses 36.5 degrees centigrade, and reactor 12, 33 degrees centigrade dynamics media. And temperature comparison in dynamics media reveals late-stage differences. Variant levels increase over time. This highlights temperature sensitivity. This slide shows non-reduced CESDS analysis when active pro media was used. We are comparing reactor 13 with 36.5 degrees centigrade and reactor 15 with the 33 degrees centigrade. Active pro media shows distinct impurity profiles. Temperature impacts fragmentation differently. These insights guide optimization. Now let's look at the reduced profiles. This slide shows the reduced analysis, CESDS analysis, and we are comparing reactor 10 and reactor 13. And reactor 10 uses dynamics media and reactor 13 uses active pro media, and both are at 36.5 degrees centigrade. Reduced CESDS confirms structural differences across media. Non-glycosylated heavy chain levels vary significantly. This links process to product quality. Non-glycosylated HC peak area increased several fold with change in media condition. This slide shows reduced CESDS analysis of reactor 10 comparing with reactor 12. And both use dynamics media here. The only change is the temperature in reactor 10 was 36.5 and in reactor 12 is 33 degrees centigrade. Temperature shifts impact reduced profiles over time. Increased variance appear at later stages. This guides process and control decisions. Non-glycosylated HC peak area increased at 12 and 14 days when temperatures changed. Here we show reduced CESDS analysis and comparison of reactor 13 and reactor 15. And these both reactors use active pro media, and reactor 13 uses 36.5 degrees centigrade and reactor 15 uses 33 degrees centigrade. Active pro media under temperature variation shows strong response. Both HC and LC levels change significantly. Monitoring enables optimization. Non-glycosylated HC decreased for 7, 10, and 12 days, and HC peak increased 2 times the area for 10, 12, and 14 days. This slide shows ICIF analysis and comparing reactor 13 and reactor 15, and both use active pro media. Reactor 13 uses 36.5 degrees centigrade, whereas reactor 5 was kept at 33 degrees centigrade. As I mentioned, both use active pro media, and ICIF analysis shows charge differences between media. Basic variants are more prominent in certain conditions. Charge profiling complements size analysis, which we have shown earlier. Higher percentage of basic variance for 7, 10, and 12 days, and lower percentage of acidic variance for 7, 10, and 12 days for reactor 13. ICIF analysis comparing reactor 10 and reactor 12. Both use dynamics media. Reactor 10 is kept at 36.5 degrees centigrade, reactor 12 at 33 degrees centigrade. And these temperatures changes impact charge heterogeneity as it is shown in the slide. Increased basic variance are observed at lower temperatures. This provides additional process insight. Here we show ICIF analysis comparing reactor 13 and reactor 15. Both use active pro media. However, reactor 13 uses 36.5 degrees centigrade and reactor 15 uses 33 degrees centigrade. In active pro media, charge remains relatively stable. Temperature has minimal impact on charge distribution. This suggests robust behavior. Temperature change do not reflect in any major changes in the percentage peak areas. TC Buster transposon technology enabled rapid development of a stable, high-producing CHOK1 cell line expressing a CD28 monoclonal antibody. AMBER 250 high-throughput bioreactors allowed efficient testing of media and temperature effects using minimal material. CESDS analysis, reduced and non-reduced, confirmed intact antibody structure with modest condition-dependent size variant changes. ICIF analysis showed media and temperature-dependent shifts in antibody charge variants during culture. Integrated upstream process development and multi-attribute characterization enabled rapid reproducible cell culture condition optimization and quality assessment. For more integrated solutions, please visit R&D Systems website. This slide shows integrated antibody development and characterization workflows. The slide summarizes the full integrated workflow. High throughput production and analytics are combined. This accelerates antibody development. Thank you for your attention. This workflow demonstrates how integrated analytics accelerate development. Morris platforms enable faster and more confident decisions.
Thank you so much, Rini, for your presentation. And now I would like to welcome back our speakers. Hello, welcome back. Thank you so much, first of all, for your great presentation and for presenting this integrated workflow that combines a lot of different technologies and spans a lot of different steps in the development workflow. So that was brilliant. So I'm going to kick off with the two letters that are on everybody's lips these days. I have no choice, AI. So Matt, you're specialized, of course, in the discovery, engineering of these recombinant antibodies. What do you think AI, in particular in machine learning, how that's going to change the work that you do, and what's going to be the impact for the discovery of future recombinant antibodies?
Yeah, thanks, Sean. Thanks for the question. Can you hear me okay?
Yes, all good.
Yeah, I mean, really, AI and machine learning, they really hold huge potential here to revolutionize antibody development. We're accumulating huge data sets here of sequence diversity, and that provides predictive power via AI to improve affinity of your antibody, as well as other behaviors like stability or developability. And so really it offers the chance to really cut down that timeline on the front end of early discovery. However, as I pointed out, that's going to generate a lot of sequences that need testing and analysis, and that's really where instruments like the Maurice can come in and provide that early developability and biophysical information that can help you select that candidate. So I think that's my answer there. Thanks, Jean-Francois, for the question.
No, thanks a lot. Well, actually, that's a great segue because I'm going to move to Srini because you're talking, Matt, about high throughput and the needs, the number of new candidates that AI will generate in the future. So in a high throughput environment, Srini, you use the Turbo CSDS cartridge for a lot of the data that you generated. And that particular person who asked the question noticed very keenly that we have two types of CSDS cartridges that you presented at the start of your talk. So why this preferential treatment for the Turbo CSDS versus the other option that we offer?
That's a great question, Jeff. The reason for that is whenever you have more samples, you can reduce the time by adapting, as the name suggests, turbo cartridge, where non-reduced run is done in 8 minutes. In contrast, if you compare the analytical CSDS plus cartridge, it takes about 35 minutes. So you save about four-fold time by just using turbo CSDS cartridge. So it depends on the samples. If you have very few samples and you wanted an analytical grade separation, you can still go with the regular CSDS plus cartridge. But if you have a lot of samples and wanted to reduce the time of analysis and still get the very quality data, you go with the turbo CSDS cartridge.
Yeah, the answer was in the name. Yeah, thank you. Jessica, moving into you. So this person has a custom gene of interest. How do they can access the TC Buster transposon backbone to use with their specific gene of interest?
Yeah, absolutely. So we have a couple clonable backbones on our website. So they either have kanamycin resistance or AMP resistance, and they just have a multiple cloning site where the ITRs are outside the multiple cloning site. So you put your own promoter, your own gene of interest, your selection gene from a mammalian selection in there. So yeah, they're just right on our website.
No, brilliant. Actually, I'm going to stick with you, Jessica, if you don't mind, because this question is related as well. A lot of the decision making to pick the viral vector of interest or non-viral vector of interest is often the size of the cargo available. Looking at these gene of interest that our audience members may have, what is the maximum cargo size that you can insert in a TC Buster?
Yeah, so the one we used today was about 12 kb total. There is technically no limit as long as you can physically get it into the backbone. The major limitation is more on the electroporator, being able to deliver that to the cells. And most of us are using transformed cell lines that are pretty hardy, so you could get a pretty good sized cassette in there into those cells.
Okay, excellent. Thanks a lot. I'm going back to Matt. So if we're looking from a therapeutic perspective this time, this person asking, why do you think that recombinant antibodies in particular have become sort of like the dominant form in the biopharma pipelines these days? You mentioned a few of the advantages, but maybe expand a little bit on that.
Yeah, great question. Thanks for that. So with recombinant antibodies, there are some real crucial advantages that I touched on earlier. I mean, you really need to be able to consistently manufacture, and a hybridoma system, for example, wouldn't offer that. With the recombinant antibody, there's also a reduced risk of immunogenicity. So these are drugs, right, that are going to be injected into a human. So that's a significant concern. And then often, these days, many of the antibody therapeutics are not your typical monoclonal, but they're really a bispecific or a trispecific or an antibody drug conjugate. And so you really need to know the sequence. You really need to be able to engineer the antibody to be amenable to those platforms. So really, it's about safety, it's about efficacy, and knowing the sequence and having it be portable as you would with a recombinant antibody really offers those advantages. And I think there's something like 900 approved antibody therapeutics right now, and they're nearly all recombinants to highlight that point. Thanks.
Thanks a lot, Matt. Srini, so salt is a critical parameter when we're doing capillary electrophoresis. Those people who are listening who do a little bit of that will surely know that. And so this person is asking for the maps that they have. So that in their example, they have 0.5 mg per mil in one time PBS, 1x PBS. What do they have to remove salts to perform CSDS analysis on the MRIs in these specific conditions?
It's a great question. As we know, salts interfere, high amount of salt interfere with the CSDS analysis. So we overcome that with a method called stacking. So the way you do it is you prepare your sample in a lower volume, and just before loading, you can add a little bit more SDS as well as the water at the final step. So you add a 1:5 ratio usually, or 1:4 ratio, in order to just dilute out the salts so that you can get a nice electrophoresis. So In that way, you can overcome even 1x PVS samples. You can just use the CES directly and then use the stacking method and overcome and you can get nice set of results. And in fact, we have used for the antibody analysis here, the stacking method. So in that way, even though the samples were low concentration, like 0.5 milligrams per ml in PBS, it can overcome those interferences.
Oh, that's very helpful. Thanks a lot, Srini. Unfortunately, we don't have time for more questions. I'm very sorry. So I'll wrap up here the webinar. First of all, I want to say a great thank you to all speakers, Matt, Jessica, Srini. Thank you very much for your presentation and for your contribution to this Q&A session. I'd like to thank you, the audience, for joining us today, whether you're watching now live or later on demand. For all the great questions that you submitted as well. And if we didn't get to answer your questions during this Q&A, of course, we'll get back to you by email, so don't worry. And at the end of the webinar, there will be a short survey popping up on your screen. If you have a few seconds, please answer that, because it's super important for us to make sure that we provide you with great content. So if you have any feedback or comments on the content itself or the format of this webinar, please let us know. That's super helpful for us for future webinars. And with that, I'll wrap up for today. Thanks again, everybody, and we'll see you on the next webinar. Have a great day.